Well, welcome to the RNA Collaborative Seminar Series from March the 13th, 2024. And we're hosted today by the RNA Therapeutics Institute of UMass Chan Medical School in Worcester, Massachusetts. My name is Jonathan Watts. I'm a professor here at the RTI. Before I introduce our speakers, just a reminder that we hope to catalyze a good discussion. So please remember to ask questions using the Q&A button at the bottom of your screen. So then without further ado, let's jump into some science. Our first speaker is Julia Alterman. Julia did her doctoral studies in Anastasia Khvorova's lab here at the RTI, and among other projects, developed the dibranched siRNA scaffold that shows widespread and potent silencing in the central nervous system. And that's the primary technology that underpins RNA Therapeutics. After a time in industry, we were lucky enough to recruit Julia back to the RTI, where she is currently assistant professor. And her research continues to be on solving the problem of delivery of oligonucleotides to extrahepatic tissues, including muscle and bone and eye, and as well, of course, the focus of today's talk, which is delivering siRNA to the skin. Julia, looking forward to your talk. Over to you.
Great. Thank you so much, John. I'll just share my slides. Okay, so I hope everyone can see these. Hello, everyone. I'm so happy to be here to talk to you about some work we've been doing on delivering siRNAs to the skin, an investigation of chemistry and routes of administration. All right, so I'll start by describing siRNAs a little bit, just so that everybody's on the same page. siRNAs, or small interfering RNAs, are these short double-stranded RNAs that once you deliver them to a cell, they get loaded into this protein called Argonaute-2. And once loaded, the sense strand, or the non-targeting strand, is discarded. And the guide strand, which has a sequence that's complementary to a target mRNA, is retained. And this now is called the RNA-induced silencing complex. And this RNA-induced silencing complex can go find target mRNA, cleave target mRNA, and prevent protein from ever being made. So you can imagine this would be an excellent treatment for a wide range of genetically defined diseases. But RNAs on their own are not stable and do not deliver well to cells without packaging, and they can have some immunogenicity. So it's really important for us to develop chemistry that allows us to use these molecules as therapeutics. And there have been decades of work in trying to develop chemistry for these molecules. And I'll show you some of this chemistry on the right-hand side of this slide here. So as you can see, these molecules have every single ribose modified at the 2' position. So whether it be with a 2' O-methyl or a 2' fluoro, you can see that every single ribose is modified. We also have terminal modifications, and these include 5' vinyl phosphonate that is protecting the 5' phosphate on the siRNAs that is essential for loading it into AGO2. And we have phosphorothioate modifications, which not only protect you against exonuclease attack, but also are a little bit sticky and so can help you with some of the delivery of these molecules. And then, of course, we can attach these molecules to conjugates. And there's a wide range of conjugates that can be used to deliver siRNAs. And so there's a big difference between these, what we call informational drugs, and your traditional small molecules. So with a traditional small molecule, the chemical structure has wrapped up in it both the dianophore, which defines kind of the distribution and PK properties of these molecules, as well as the pharmacophore, which gives it its targeting properties. And so the thing about a small molecule, though, is if you try to adjust it to change one of these two things, you're likely going to impact and change the other. This is not true for informational drugs. With informational drugs like siRNAs, we can actually design chemistry and conjugates that give you delivery to a particular organ of interest, that give you stability and long-term duration of effect, all of these PK properties. And then we can plug in different pharmacophores or sequences to target different genes. And so what this means essentially is that once you have designed a scaffold that delivers well to an organ of interest, you can target many different diseases by simply switching out that sequence to target a different mRNA. And this has been done. There's actually 5 approved drugs on the market that are using unformulated conjugated siRNAs. And they are all attached to this ligand called GalNAc, which is specific to a receptor on the surface of hepatocytes. And so these GalNAc siRNAs deliver beautifully to the liver. And as you can see in the graph on the bottom right, actually after a single injection of these GalNAc conjugated siRNAs, you can get long-term duration of effect of these molecules out to 6 months to a year. So this means that patients can be dosed very infrequently and benefit from long-term silencing of the problem gene. So in our lab, we're actually really interested in trying to figure out how we can use these molecules outside of the liver for extrahepatic delivery. And so we're working on projects trying to deliver siRNAs to the heart, placenta, lungs, brain, muscle, kidney, and skin. And we're doing this by using different kinds of conjugates as well as different kinds of structures. So we have a library of hydrophobic conjugates. We have fatty acids, sterols, vitamins. And while none of these are truly specific the way that GalNAc is, so this is not receptor-specific targeting or cell type-specific targeting, they are sort of tissue-enhanced delivery, have a tissue-enhanced delivery aspect to them. And what that means is that some of these conjugates will go more to some tissues and others will go more to other tissues. And on the other side of this, we also have multivalency. So we can make these molecules as a single siRNA or as a dimer, a trimer, and a tetramer. And these structures now, with their change in size, also have different and unique delivery properties. And we also have another conjugate that I'll probably talk a little bit more about today, which is an amphiphilic conjugate. So this has got some hydrophobic pieces to it, as well as some hydrophilic pieces to it. So this chemistry really can give you a broad range of the kinds of organs, tissues that you can target. And even these molecules are moving towards clinic. So the divalent molecule that I just described to you that was developed at UMass Medical School is now being used by Andrei Therapeutics to target a number of neurodegenerative diseases in the brain. And one of our hydrophobic conjugates, also developed at UMass Medical School, is being used to treat preeclampsia by delivering it to the placenta. And so this is really exciting. This means that all different kinds of architectures of siRNA are being moved towards clinic, and we're really breaking open the body and looking at all of these different organs that we're going to be able to target in the future. But today, I'm particularly interested in talking to you about some of the work we're doing in the skin. So skin not only is the largest organ in your body, but also skin diseases are some of the most common. So they affect more than 900 million people in the world at any given time. And so to do these studies, we need a target to go after first. And the target that we chose to use was JAK1. And JAK1 is a target that is involved in multiple different inflammatory processes, and therefore is actually implicated in a number of different skin conditions. And two of these conditions, atopic dermatitis and alopecia areata, affect more than 30 million people in the US alone. And so first, you need to actually find a sequence that targets JAK1 well. So we did a screen of JAK1 siRNAs, both against the human mRNA for JAK1, as well as for the mouse JAK1 mRNA. And we were able to find multiple hits. And in fact, we were able to find one hit that was cross-homologous between the two. So this means that we can use the exact same siRNA to target both human and mouse. And if you look on the right, you can see that this cross-homologous JAK1 compound, numbered 3033, gives you beautiful mRNA silencing in both human and mouse cells, as well as nice protein silencing in human and mouse cells. So we know that this is a sequence that can work in both of these systems. We also wanted to look at the selectivity of this compound. So you want to make sure, of course, that your compound is only going to be hitting JAK1 and not any other of the JAK family compounds, or JAK family genes, sorry. And so as you can see here, JAK1 3033 silences just JAK1 and has no impact on JAK2, JAK3, or TYK2, showing that it really is specific to JAK1. And this makes sense because we designed the sequence to be specific to just this gene. So we also have to think about how we're going to deliver these compounds to the skin. So the skin actually, even though it's, right out there on the outside and seems like it would be easy to deliver a drug to the skin, it is not, in fact, easy to do so. Your skin is covered by something called the stratum corneum barrier, and this barrier is meant to protect you from large molecules like siRNAs penetrating into the skin and getting into your body. And so how do we get past this stratum corneum barrier? Well, there are two different routes of administration that we explored to look at how we could overcome this barrier. One is direct intradermal injection. So this means just simply injecting the siRNA directly into the dermis. A second one was using star particles for transdermal, or essentially topical administration of siRNAs. So these small star particles are ceramic microstars with about 500 micron arms. And when formulated, you can rub them on the skin and they will make small perforations in the skin that will allow larger molecules like siRNAs to penetrate deeper into the skin so that you can get a silencing effect. And these were developed at Georgia Tech. So the conjugate chemistry that we decided to use for these initial studies, so we picked two different conjugates that we wanted to look at, each with their own chemical properties. We picked DCA, which is a hydrophobic conjugate, and we picked dendrimer, which is an amphiphilic conjugate, and you can see those pictured here on the right. Okay, so first I'm going to show you some data from intradermal delivery. So when we inject these siRNAs directly into human ex vivo skin, we wanted to see what cell types it accumulated in. So we injected unconjugated JAK1, so this is the siRNA with no conjugate attached, as well as DCA conjugated JAK1 and dendrimer conjugated JAK1. And as you can see, all three actually show accumulation in multiple different cell types in the skin. So you can see accumulation in immune cells, fibroblasts, endothelial cells, keratinocytes, and monocytes, all to varying degrees. And you can see that it looks like the DCA JAK1 may be accumulating slightly more than the other two conjugates. And this makes sense since it is a hydrophobic conjugate and potentially a little bit more sticky. And we can also see that this resulted in significant silencing in ex vivo human skin, again, with all three conjugates, but with DCA showing us potentially the best silencing. And this is both in the epidermis as well as in the dermis. So the epidermis is the sort of top layer of the skin below the stratum corneum barrier, and the dermis is just below that. We also wanted to know whether or not we were having an impact on any downstream signaling molecules. So to do this study, what we did was we injected JAK1 siRNA, again, into human ex vivo skin, and then we stimulated with interferon gamma, which should lead to the expression of chemokines like CXCL9, CXCL10, and CXCL11. And we wanted to know whether or not our JAK1 siRNA could block the expression of these chemokines. And as you can see here in the graphs below, we were able to show significant reduction in CXCL9, CXCL10, and CXCL11 as a result of treating the skin with DCA conjugated JAK1 siRNA, but not from PBS or non-targeting control treated oligos. Another concern when thinking about delivering molecules that regulate the immune system is you really don't want to have an overall whole body dysregulation of the immune system. We really want to make sure that we're just targeting the organ of interest so that there aren't any safety concerns. And so what we wanted to do, we went into a live pig and we wanted to see if we do intradermal injections into a live pig, will we be able to see any accumulation of this oligo in other organs throughout the body as it gets cleared? And so we did that. We did about 40 injections into a live pig of these oligos for a total of 5.2 milligrams injected dose. And we looked at the accumulation of this siRNA both in skin as well as in liver, kidney, spleen, and heart. And as you can see here, the majority of the oligo is in fact retained and found in the skin, while very little goes to the liver and undetectable amounts, or if any, go to kidney, spleen, and heart. So it's about 15 times, at least 15 times more accumulation in the skin over clearance organs. So this is great. This means that potentially we could use this method to have skin-specific delivery without having a skin-specific conjugate. So now I'm going to move on to star particles. So these are some images from the paper from the people that developed star particles over at Georgia Tech. And so you get to see a little bit more what these look like close up. You can see them on a person's finger down in the bottom left corner of the slide. They're quite small. As I mentioned, they're ceramic. And when you rub them onto pig skin, as you can see here on the right, stained with gentian violet, you can see that they've made small perforations throughout the skin. And these will be the tiny holes that siRNA will be able to penetrate to. And so there actually have been a few studies done with these star particles looking at how well they're tolerated by patients. You know, people always ask, are they going to hurt? A study was done in patients asking them how comfortable or uncomfortable they felt star particle application was relative to the use of a hypodermic needle. And so you can see here that 92% of patients said that the star particles felt comfortable, while only 8% said that they felt uncomfortable. And this is different from the hypodermic needle, where 58% of the patients said that it felt comfortable and 42% said that it felt uncomfortable. They also, the authors of this study also wanted to look at how familiar people got over time with using these products. So the first question was asked, how comfortable or uncomfortable would you feel applying these skin products on your own? In the beginning, the majority of people felt comfortable, and then the rest were split between very comfortable and neither comfortable nor uncomfortable. After 10 days, the amount of people that felt very comfortable increased, and the amount of people that felt just comfortable decreased. And so it seems that you can become more comfortable with using these star particles over time. It was also asked how people thought these compared to their regular everyday skin products, so things like body lotion or sunscreen or face wash. And they were asked if it was similar or if it was different. And so on day one, when they were asked, many people actually felt that it was very different to their typical skin products, and only a few said that it felt similar. But you can see on the right that many more people said that it felt at least very similar, similar, or neither similar or different. 50% of people, while 50% still felt that it was different. So as you use these products over time, you become much more familiar with them, and the ease of using them grows. And so here are our studies, the setup for our studies with star particles. So what we did was we formulated our star particles in aloe vera, and then we rubbed it on ex vivo human skin, you can see here in the middle, using approximately 40 kilopascals of pressure. And then we treated them with siRNAs. And to do this, we actually glued a little bit of tubing to the top of the skin and then put the siRNA inside of that tubing. And we did this to ensure that none of the siRNA would leak in from the bottom of the skin, and that we would just be treating from the side of the stratum corneum barrier. And what we were able to see was that we could get accumulation in all the same cell types as we did with intradermal administration with these star particles. So again, you can see it in immune cells, fibroblasts, endothelial cells, keratinocytes, and melanocytes. And that this does result in significant silencing of JAK in human skin with star particle treatment. And as a control here, we also had no stars. And so for this, we just rubbed aloe vera and then treated the skin with siRNA as shown in the previous slide. And you can see that without the star particles, we get no penetration and no silencing with these siRNAs. But with the star particles, we are able to see significant silencing both in the epidermis as well as deeper in the dermis. And lastly, and I think this is the most exciting study, we actually brought these star particles into live pigs. And so for this study, what we did was we actually formulated the siRNA in excipient with star particles. So instead of pre-treating with the star particles and then applying the siRNA afterwards, we formulated everything into one gel or formulation. And that gel or formulation was rubbed on the surface of the pig skin. And then 4 days later, we looked at whether or not there was silencing. And we were, in fact, able to see significant silencing in a live animal, a live pig, with essentially topical or transdermal delivery of siRNAs. And so this was really exciting, and I think it opens up a whole new area that we can use these small interfering siRNAs in. And yeah, with that, I will conclude and wrap up here. siRNAs can be used to silence JAK1 in both human skin ex vivo and pig skin in vivo. And siRNAs can be delivered intradermally with minimal liver exposure. And transdermal delivery offers a less invasive method for delivery of siRNAs to the skin. Moving forward, we're interested in looking at a lot of new targets, as I mentioned. You can essentially design siRNAs to target any gene and plug it back into your scaffold of interest. We are also working on novel conjugation strategies to see if we can improve the delivery of these siRNAs to the skin using this star particle system. And this is all wrapped up in sort of optimizing this siRNA star particle delivery system. So with that, I would like to thank everybody who has been involved in the project over the years, all of the chemistry that's been developed by the Khvorova lab. But a few names in particular that I would like to point out are Dr. Anastasia Khvorova, Dr. John Harris, Chi Tang, Hassan Faki, Zainal Abedin, and Katie Gross, who were all instrumental in developing these star particle assays and doing these skin studies. And this work was funded by RNA Therapeutics. And so they're our partner in developing these for clinical applications. And so with that, I would be happy to take your questions.
Excellent. Thank you, Julia. Really great research. So there are a couple of easy, potentially quite short questions that we can deal with first, and then a couple that may be more open-ended. So what was the source of the ex vivo human skin that you used?
Well, we have two sources. One source was actually through John Harris. And this comes from patients who have lost a lot of weight and need to get some skin removed. And then we get access to that skin and we can use it for experiments. If we do not have access through that, you can actually order human skin online.
Great. The next couple of questions that maybe we can deal with are related to the modification pattern of siRNAs. So there are kind of three related questions. Two first from Rajur, is every nucleoside modified in a given sequence, and are all the positions modified? Maybe you can explain. He asked 2', 3', 5', but you can clarify there. And then a follow-on question from Lakshmi Bhagat, will Mo modification at 2' be tolerated instead of 2' O methyl?
Yeah, great questions. So the chemistry of our molecules is every ribose is modified at the 2' position with either a 2' O methyl or a 2' fluoro. We have different patterns of these modifications, so sometimes they'll be in different positions, but it's always just a 2' methyl or a 2' fluoro. We also protect all of our ends with phosphorothioates, so every end has at least 2 phosphorothioates on it, except for our single-stranded overhanging tail, which has 7 phosphorothioates on it. And then, of course, we have our conjugate. I do not know about the tolerability of Mo in siRNA. It may be tolerated at some positions. I think probably John could answer that question better than I could, but we haven't tried it ourselves in the context of these siRNAs.
There were some early studies on that. You're right, Julia, and Lakshmi too, that found it wasn't as easy to use in siRNAs as 2' methyl is. So that's tended to be the focus. Great, thanks, Julia. How often do you need to inject the siRNA intradermally? And do you inject only at one site or multiple sites?
Yeah, great question. So we haven't really been able to do duration of effect studies yet because the human skin only lives so long in culture. So all of the studies I've shown you here today have been about 4 days long, which is about as long as we can keep it in culture. But the plan is to actually get some pigs so we can test exactly that. We want to know how long is this silencing going to last, especially if you decide to go with an intradermal route of administration. You really would like to be able to treat the patients as infrequently as possible with intradermal injections, since they have to come into the doctor's office for that, and it may be irritating. So, the hope is that with all of the new chemistry that we have and the ability we've had to stabilize these molecules, we're hoping to be able to get at least a few weeks, if not months, duration of effect. But those studies still have to be done.
Thanks very much. Great. There's a question from an anonymous attendee that says, great talk. Have any ligand determinants of siRNA targeting to the skin for uptake and distribution been identified, or is it solely ensured by the route of administration, such as star particles?
Yeah, that's a great question. And we don't have anything yet, but we are looking into that. Obviously, you can imagine that Another way to bypass kind of treating the whole body with JAK1 and just treating the skin with JAK1 would be to have a skin-specific ligand for delivery, and then doing sub-Q injections would be so much easier than having to treat patches of skin, especially in the case of an intradermal injection where you're going to have to do lots of intradermal injections in order to cover even a small area of skin. So we're looking into it. That would be a great next step. I think in between having the star particles and being able to kind of rub over a larger area, maybe something in between that can work in the meantime.
Great, thanks very much. Building on this question of conjugated siRNAs in skin, at the bottom of the question list here, there's one from Supriya Day that says the dendrimer conjugate was not very good, not as good as the other conjugate. Does lipophilicity matter? Is that a key constraint?
Yeah, so we think that lipophilicity might matter. I'm still curious to see in other studies when we move into large pigs whether or not this stands. The idea behind using some of the branched molecules or the amphiphilic molecule was that maybe if you had a compound that was a little bit less sticky, but that could still get taken into cells, you might have better spread. And so you would have to do fewer injections as you could spread them further apart from one another. So this was kind of the rationale for trying these two types of conjugates. It seemed in these studies that DCA was certainly performing better. And we know that hydrophobic conjugates are a lot more sticky. So if you're looking right at the site of injection, those are likely to give you the best effect. But there may be some play in there where we might be able to walk back the hydrophobicity a little bit to see if we can get a little bit more distribution and spread and cover a larger area. So it's certainly something that needs to be played with.
Great. And then there's one more that's sort of related to conjugation. Rahul Agrawal says, were unconjugated versus conjugated siRNA, did they show similar responses on the JAK pathway? And I'm guessing that might have been for comparable levels of gene silencing, if those were able to be done.
Yeah, so is the question whether or not unconjugated and dendrimer showed similar effect on downstream signaling, or just in silencing in general?
Maybe you can answer both as far as
I can answer both.
Yeah.
Okay, yes, they did show similar levels of silencing. Again, local administration is very different from systemic administration. So when you deliver heavily chemically modified oligos locally, and those oligos have a fully phosphorothioated tail, they absolutely do get taken into the cells that they are exposed to. It's sort of when you do things more systemically or need distribution to a larger area where those sort of smaller unconjugated oligos don't work as well. But for local skin administration in an 8 millimeter punch, unconjugated was working quite well, and you saw it look just as good as the dendrimer conjugate. I believe we have just been trying to run these studies looking at downstream for all three of these conjugates, and I think dendrimer may be performing a little bit better on being able to lower the downstream chemokines, but neither of them perform like DCA.
Great, thanks very much. There are a couple of other questions there, but I think we'd better move on to Andrei's talk. So perhaps Julia can answer the remaining questions in written form. Thank you all for the good questions.
Great, thank you all.
All right, and thank you, Julia. So our next speaker for this collaborative seminar is Andrei Korostelev, who is professor at the RTI. His interest in the ribosome and its structure go back to his postdoctoral work with Harry Noller at UC Santa Cruz. And he set up his independent lab here at the RNA Therapeutics Institute in 2010. And since then, he's been using ensembles of cryo-EM structures. I always think of them almost a bit like atomic resolution movies, to uncover really wonderful mechanistic insights, including mechanisms of translation termination, of ribosomal assembly and dynamics, of fidelity, of frame shifting, and so on. And today he'll speak to us about the molecular mechanism of angiogenin activation by the ribosome. So over to you, Andrei. Thanks very much.
Thank you very much, John. It's a pleasure to present our recent study that aimed on understanding the molecular mechanism of activation of angiogenin. Angiogenin is a also known as RNase 5. It's a small 123 amino acid long ribonuclease that belongs to the family of RNase A enzymes. This protein was discovered by the Valley lab as an angiogenesis factor. It is now known to be activated during stress, for example, hypoxia, which signals that new blood vessels must be grown. It is not surprising, therefore, that angiogenin is upregulated in cancers, which rely on angiogenesis to provide increased food and oxygen supplies. Another prominent role of angiogenin is as a neuroprotection factor. Consistent with this role, mutations were found in sporadic and familial amyotrophic lateral sclerosis, Parkinson, and Alzheimer's diseases. And finally, in more recent studies, angiogenin has emerged as an epigenetic inheritance factor. Specifically, it was found to produce tRNA fragments, which can be transmitted to offspring via sperm, and were found to mediate various metabolic responses depending on paternal experience, such as diet, stress, and so on. tRNA is the primary target of angiogenin. The cleavage occurs in the anticodon loop. So the products represent the so-called tRNA halves that are about 30 to 40 nucleotides long. And they're also termed tRNA or tsRNAs. Most pronounced downstream effects of tRNA cleavage are on translation, which is not too surprising considering that in the first place, tRNA cleavage can lead to the depletion of the pool of translationally active tRNAs. In addition, there are some specific effects, such as inhibition of translation initiation by certain fragments of tRNA, such as tRNA ALA or tRNA cis. What remained an enigma in the field for a while is how angiogenin is activated. It captured the attention of biochemists that the catalytic activity of angiogenin in vitro is several orders of magnitude lower than that of RNase A and other homologs, which, as you know, are very efficient enzymes. This graph of pH titration, for example, shows that the activity in one study for angiogenin was found to be about 6 orders of magnitude lower than RNase A. In practical terms, it means that if you'd like to observe tRNA cleavage by isolated, purified angiogenin in vitro, you would need to incubate micromolar concentrations of angiogenin, which is way too high for an enzyme, with high tRNA concentrations or over a long time. In contrast to RNase A, which, as you know, shreds RNA very quickly at nanomolar concentrations. By quickly, I mean in seconds. So the enzyme was pronounced to be very weak. The enzymatic activity is limited, and it was nevertheless stated that the weak ribonucleolytic activity is critical for its biological functions. This finding was corroborated by crystal structures. The folds of angiogenin and RNase A enzymes are very similar, featuring similar positions of the catalytic residues that you can see in these images, 2 histidines and lysine. As you can see, in RNase A, the substrate analog in that study was well accommodated in the active site, with the C-terminus of the enzyme pointing away from the substrate. So one notable difference in the crystal structure of angiogenin was that its C-terminus forms a short helix. And if we look at the surface view of the structure, you will notice that the tail occludes the active site. So it would clash with the binding of the nucleotide, consistent with poor Michaelis constants for this enzyme. Now, what remained intriguing was that many residues whose mutations were identified in neurodegenerative diseases and were thought to lead to loss of function, as I mentioned before, are located on the surface opposite from the active site, which in this view is in the back. These observations suggested to us that there may be a modulator of activity that angiogenin interacts with. In fact, it's been known that angiogenin in cells or lysates is very efficient, as nanomolar concentrations of angiogenin can induce tRNA cleavage very quickly. Furthermore, Valli and colleagues have shown in their foundational study that the biological activity of angiogenin is very high. And they presciently noted in the manuscript that a cofactor may exist that activates angiogenin, and in this case, the binding affinity specificity would have to be quite extraordinary. So we thought to explore how the RNase of angiogenin is activated and how the extraordinary specificity toward tRNAs can be achieved. Dr. Anna Loveland undertook this adventure and performed a battery of experiments that helped us to answer these questions. Our work started with using translationally active lysate, in this case, rabbit reticulocyte lysate, to test the translational repression by angiogenin. Consistent with previous findings from many other labs, angiogenin potently inhibits translation in the 30-ish nanomolar range. Although Anna tested different batches of RRL showing a range of translational activities, likely due to different concentrations of components, all preparations are completely repressed by about 70 nanomolar angiogenin. And what was observed here is a canonical luciferase translation over time. Now, what was also consistent with previous studies was that inhibition is relieved by the addition of RNasin, which is a specific inhibitor of RNase A nucleases. And this protein has picomolar affinity to RNase A enzymes that in which the binding occludes the active site. So this assay showed that translation inhibition is indeed due to the nuclease activity of RNase. So consistent with this finding and previous work, we thought that cellular lysates might contain the activator of angiogenin. Anna took the RRL angiogenin mixture and fractionated it over sucrose cushion to see whether angiogenin would associate with the lighter components of the lysate or with the heavier ribosome pellet. And the Western blot showed that angiogenin co-sediments with the ribosome pellet. This co-sedimentation is inhibited by RNasin, suggesting to us that the activity of angiogenin in the absence of RNasin may be dependent on the enzyme's ability to bind the heavier fraction, which is the ribosome, and the binding could be direct or indirect. So we decided to explore if angiogenin directly binds the ribosome. To this end, we used the ribosomes from RRL in the presence of angiogenin, and Anna subjected these ribosomes to cryogenic electron microscopy and determined a 2.8 angstrom structure. This work found angiogenin in a very prominent class at high abundance, bound to the ribosome in the A site of the small subunit. It was interesting that despite the presence of many different states of the ribosome, as is often the case, different rotational states and tRNA occupancy states, angiogenin is sitting in this elongation-like conformation of the ribosome and occludes the A site. When we move closer into the A site, we will see that the protein is very nicely fitting into the A site, and it's surrounded by the universally conserved and key components of the ribosome, including helix 69 of the large subunit, helix 44 of the small subunit, where the decoding center is located, the head of the small subunit, and P-site tRNA, as well as mRNA. Well, this view shows the density in which the side chains of angiogenin are very nicely resolved, interacting with pRNA and ptRNA and mRNA. And another interesting fact is that this binding buries more than 2,000 angstrom squares of angiogenin, or 30% of total surface area, which further indicates that the binding must be pretty strong and specific. The sequence alignment with RNase A underlines that angiogenin is quite unique. In this alignment, you can see that colored residues, which correspond to the region of the ribosome with which they interact. In this case, the blue ones correspond to the larger ribosomal subunit, and the blue ones correspond, yes, large, and the yellow ones are the small subunit, and the green ones are mRNA. The catalytic residues are shown in magenta. So what you will see that residues interacting with the ribosome, 65% of them are unique to angiogenin compared to RNase A. And they're often aromatic, hydrophobic, or positively charged, so that they could interact with the ribosomal RNA. For example, in this case, you can see the K54, which is glutamine in RNase A, or the MKR motif, which is a PLV. Now, what's even more interesting is that the circular residues correspond to those whose mutations were found to result in loss of function variants associated with neurodegenerative diseases. 60% of them interact with the ribosome. So it was exciting to see that angiogenin can bind the ribosome, but does it tell us anything about the mechanism? Now, what we recognized immediately is that angiogenin changes its conformation, and its C-terminal tail is now a beta strand, which is very similar to the structure of RNase A. And this conformation is stabilized by the interactions with the ribosomal RNA helix, protein S15, and P-side tRNA. These interactions explain why the alpha helix is rearranged into a beta strand. Furthermore, the alpha helix would clash with these elements of the ribosome, and the canonical isolated angiogenin conformation would be incompatible with the ribosome. It was also very suggestive of the mechanism that the active site residues, shown here as gray spheres, are oriented toward the intersubunit space at the P-stalk area, where tRNAs are normally delivered to the A site of the ribosome during translation. So we asked, is ribosome-bound angiogenin efficient in cleaving tRNAs? Anna incubated 10 nanomolar free angiogenin with tRNA alum, which is one of the bona fide substrates of angiogenin, like in previous studies, and she observed no cleavage in 100 minutes, consistent with the studies showing that angiogenin is a very poor enzyme. When she added the ribosome, then tRNA cleavage was very efficient, with fragments detectable, as you can see in this gel, even in 1 minute after incubating of angiogenin with the ribosome. When she added RNasin, then this cleavage was inhibited, and it is pretty easy to see why that is the case. So when you look at this complex of angiogenin with RNasin, shown here as a blue structure, which is a horseshoe-like protein that surrounds angiogenin's active site, this structure is incompatible with binding to the ribosome, as RNasin would clash with the small ribosomal subunit. So that explains why angiogenin is not activated. As you know, in the cells, most tRNAs are aminoacylated and delivered to the ribosome, not as deacyl tRNAs alone, but by elongation factor 1A. So we next tested if this complex can be a substrate for the ribosome-bound angiogenin. This gel shows that in the presence of elongation factor 1, tRNA is cleaved even more efficiently. In 1 minute, you can see accumulation of the fragments that corresponds to more than about half of the reaction. So our estimate based on these findings is that tRNA cleavage in this complex is at least 1,000-fold faster in the presence of the ribosome than in the absence. Finally, Anna thought to see if tRNA can be captured together with the ribosome-bound angiogenin. She formed a complex and did a cryo study, and the complex contained a EF1A bound with non-hydrolyzable GTP analog and amino-acylated tRNA. She observed, as expected, that tRNA is bound to the ribosome with its anticodon loop stuck right into the active site of angiogenin. Since this complex was incubated longer than several minutes, it is not surprising to see that the anticodon loop is very disordered, meaning that we do not see density that would cover that region. This is consistent with angiogenin nicking the tRNA, resulting in the splitting of the anticodon loop. In addition, she also found that tRNA can bind to the ribosome and angiogenin in the absence of elongation factor 1, consistent with the finding that the protein cleaves deacyl tRNA pretty efficiently. By now, we were pretty convinced that the ribosome is the long-sought activator of angiogenin. However, as a critical reviewer might say, what if these results are due to non-specific sticking of a positively charged protein to the negatively charged ribosome? So we asked if a K9454E mutation could answer this question. This mutation is associated with sporadic ALS and was shown to not affect the overall structure of angiogenin and its very low catalytic activity in isolation. This is not surprising because, as you see in this picture, K54E, which is part of alpha-3, is located on the surface that's opposite to the active site of angiogenin. And this view shows that in our structures, in our cryo maps, K54 interacts with the RNA backbone of the large subunit RNA. So we thought that charge reversal would be responsible for the loss of function on the ribosome. This is indeed what Anna observed when she separated the ribosome and light fractions of the rabbit reticulocyte lysate. K54 is not present in the ribosome pellet, which contrasts the other mutant that she characterized. That's histidine 13A, one of the catalytically dead mutants. And since the catalytic residue helix histidine 13 is exposed in solution and does not contact the ribosome, it is not surprising that this mutation does not perturb ribosome binding. Now, when she performed the tRNA cleavage assay, no activation was observed in the absence or presence of the ribosome for K54E, as well as for the control mutant H13A. When these mutants were then added to the translating lysate, their inhibitory activity was more than an order of magnitude worse than that of wild-type angiogenin. So our results show that the ribosome binding is required for activation of angiogenin RNase, tRNA cleavage, and translation inhibition. Based on these findings, together with many previous studies on translation and angiogenin, we can propose how angiogenin is activated during stress. Under normal conditions, it would be disastrous for a cell if tRNAs can be cleaved by angiogenin. So angiogenin is primarily kept in check by two mechanisms. First, it is bound with angiogenin inhibitor, which severely limits the amount of free angiogenin that can be activated. Second, as we know from numerous studies, actively translating ribosomes often contain their A site occupied primarily by amino-acylated tRNA that either enters the A site with elongation factor 1 or enters the A site without EF1 or then translocates or pre-translocates to the P site when it's bound to the peptide. So active ribosomes are not consistent with angiogenin binding. Under stress or in particular signaling pathways or during disease, angiogenin may bind the A site because both these pathways would be affected. First, angiogenin inhibitor is known to be downregulated transcriptionally as well as is degraded through ubiquitination. So angiogenin is now released. However, its C-terminus is in an alpha helical conformation, so angiogenin is still inactive. At the same time, cellular stress may cause ribosomal stalling, which was shown by previous cryo-EM and ribosomal profiling and other studies, results in accumulation of ribosomes whose A site is vacant. So this combination results in angiogenin binding to the A site, activation of the catalytic center, and cleavage of tRNAs, which then play roles in translation inhibition or reprogramming, regulation of apoptosis, angiogenesis, neuroprotection, and many other processes. It remains to be shown whether the nicked tRNA can be actively unwound so that separated tRNA halves could play these roles, or whether nicked tRNA itself can play roles in these processes. With that, I would like to thank Anna Loveland, who's done this amazing work, and the lab from whom we received numerous feedback during this process. Also, I would like to thank Alan Jacobson and Robin and Chasan, whose thoughts and materials we used, and it was a great collaboration. Also, I would like to thank our cryo-EM center with Chen Zhu directing the facility and doing wonderful work in collecting and storing the data. And thank you very much for your attention.
Excellent. That was a wonderful, wonderful talk, Andrei. Thanks so much. There are several questions in the Q&A. First of all, two questions from our chair, Phil Zamor. He asks, are the cleaved tRNA halves released, or does angiogenin have to presumably depart from the ribosome first?
That's a great question. In our assays, we can observe multiple turnover cleavage. So nicked tRNAs apparently can leave the A site, but we do not know whether this is coincident with angiogenin departure. So we're looking into this now.
Great. And then a follow-up question, does angiogenin only efficiently cleave tRNA with a peptide attached? Or can it be any tRNA?
Well, as the experiments that I showed in the beginning demonstrate, deacyl tRNA can be an efficient substrate. Now, peptidyl tRNA is that this person thinks about is probably not a substrate, because peptidyl tRNA would be occupying the ribosomal either A or P site. So in this case, angiogenin would either be unable to bind to the A site, or if peptidyl tRNA is in the P site, angiogenin in the A site would not be able to reach to the P site tRNA. It only cleaves tRNAs that are delivered to the ribosome from the A site.
That's fascinating. Fabrice Leclerc asks, how likely is it for the C-terminal helix to adopt a beta sheet? Is that transition from an alpha helix to a beta sheet observable when the protein is in solution by itself?
To my knowledge, there are no studies reporting that this transition is observable. It is most likely a very inefficiently formed conformation in solution that gets stabilized by the ribosome. But there are mutational studies which showed that mutations of this alpha helical tail that probably disrupt the alpha helical conformation increase the activity of angiogenin up to 30-fold.
Excellent, thanks. And then Mark Gray asks, great talk, are the tRNA modifications present in the anticodon loop affecting tRNA cleavage?
That's a great question, and I cannot answer this because we have not studied this topic. I suspect that they might, because they might affect the conformational dynamics of tRNAs. But I cannot give you a particular answer about particular modifications.
Great. Our own Zhoshang Xu asks, how does angiogenin loss of function lead to motor neuron degeneration?
How
does loss of function of angiogenin in these ALS-associated mutations, how does that then trigger degeneration of motor neurons?
That's, to my knowledge, is an active area of studies. It is not quite clear how angiogenin plays its neuroprotective roles. So I do not think that I can answer this question.
Fine. The next question asks, do you see angiogenin cleaving mRNA in the ribosome or only transfer RNAs?
We do not have evidence of angiogenin cleaving mRNA, as to my knowledge is the case in other studies as well, and our structure suggests that it would be impossible for angiogenin to cleave the mRNA because it actually kind of blocks the A site so that the mRNA would be unable to leave the A site and enter the angiogenin's active site. However, it is something that should be explored in the future.
Great. And then sticking to the substrate specificity question, is there tRNA specificity of cleavage? Lexi Wolfson.
Well, there is nucleotide specificity that's well characterized. So angiogenin prefers a particular purine-pyrimidine sequence, but so that would affect the specificity toward tRNAs. So that's one aspect. Another aspect is the conformational flexibility of tRNA that might affect it. So the answer is yes.
Great. And then jumping down to the bottom of this list, there are studies suggesting that angiogenin may cleave the TD loop. Does your structure support that?
They do not, the structures do not support that. However, it is not impossible for certain RNAs to bump into the angiogenin's active sites stochastically. So I cannot exclude some non-specific cleavage.
Great. And then the last question from this Q&A list, are there any alternative cell type specific inactivators of angiogenin known?
Alternative to RNase inhibitor? Not to my knowledge.
Good question. Yeah. The RNase inhibitor is fascinating. It was fascinating when you showed your RNase in structure bound. It was not obvious to me which binding interaction would actually have been more, higher affinity based on the depth of sitting in the ribosome of this angiogenin, plus of course the structural, divergence compared to other RNases. Is that a result that you would have been able to confidently predict ahead of time, or was that a surprise to you?
Well, it's very hard to predict binding affinities from structures.
Sure.
I mean, one can predict that, but the question is whether they will be true. So It is known that the affinity of angiogenin to RNase inhibitor is picomolar. So you really need to degrade that protein in order to free angiogenin. And it's unlikely to be the case for angiogenins binding to the ribosome. So we estimate that the affinity is on the order of nanomolar. That's probably similar to regular ribosomal substrates, which usually bind with single digit or tens of nanomolar affinities.
Great. Well, thank you all for your questions. And thank you again so much, Andrei and Julia as well for outstanding talks.
Thank you.
Bye for now, everybody.
Thank you.